additional complexities in the process of transcription. Then there are three.
Yes, continue.
There are three RNA polymers in the nucleus: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Then the primary transcriptions or RNA in the eukaryote contain exons and introns. And the primary transcript has to undergo the process of splicing, where the introns are removed and the exons are joined together. In the post-transcription modification or processing, it has three parts. The first one is splicing, where it is a process where the removal of introns and the joining of exons takes place. And then the second one is capping, and third one is tailing. Then,
Yes. What happens in capping and tailing?
We put caps in the RNA.
Yeah. What are the caps? What is the chemical nature of the cap?
MGPP.
Yes. 3-methyl guanosine triphosphate. Okay. And what happens in tailing? Meenakshi?
The polydylation.
Yes. Multiple adenylation. Yes. Continue.
Then we studied about genetic code. There are 20 amino acids, and among them, 3 are aromatic amino acids. First one is phenylalanine, second one is tyrosine, and the third one is tryptophan. Then the start codon is methionine, AUG.
Yes.
Then there are 3 stop codons. Then we study about translation. It is the process of polymerization of amino acids.
In genetic code, we studied about a lot of other things as well. You see, there are 6 points that we studied about. You don't skip all these points. What did we learn from genetic code?
The codon, there are 61 codons for amino acids, and 3 codons do not code for amino acids, and they function as stop codons.
Yes.
And some amino acids are coded for more than 1 codon, hence the code is degenerate.
Right.
The code is read in mRNA in the conjugate fashion. These are no punctuation. There are no punctuations. The code is nearly universal, means from the bacteria to human, the coding is almost similar. And AUG has dual function. It codes for methionine, and it also acts as initiator codon, also known as start codon. There are 3 stop codons: the UAA, UAG, and UGA. Then we study about translation. Translation, it is the process of polymerization of amino acids to form proteins based on the mRNA sequence. The bond between the 2 amino acids are called peptide bond. Then the rRNA, the ribosomal RNA. The ribosomes are made of RNA and protein. There are 2 types of ribosomes, bigger subunit and
there are not 2 types of ribosomes. They are the 2 subunits of ribosome.
2 subunits of ribosome, bigger subunit and smaller subunit.
Yes, together they make the ribosome.
Yes. Then for tRNA is in L shape for 2D, and it is claw shaped in 3D. And it is an adapter molecule. It was earlier known as sRNA or soluble RNA. And then it has tRNA adapter molecule has a 3-pair amino acid acceptor site and anticodon loop, which contains complementary base to the code of the mRNA.
Correct.
Then tRNA is specific for every amino acid. there is a special tRNA known as initiator tRNA that carries methionine, the start codon.
Yes.
And then we start about charging of tRNA, the activation of amino acid in the presence of ATP during the linkage of their specific tRNA is known as charging of tRNA, or it is also known as amino acylation of tRNA. And if two such charged tRNA comes close together, then the formation of peptide bond between the amino acids that they are carrying takes place.
Exactly. Okay. Thank you so much, Minashi. And others who have joined just now, good evening. And if you have any doubts, please ask before I begin today's topic. So today we'll be starting with the translation, like the process of translation or the mechanism of translation. Anything you want to ask me before that? So you have understood charging of tRNA, about the tRNA, ribosome, its two subunits, and etc. Right? So everything is clear?
Yes, sir.
Yeah. Now the actual mechanism of translation begins when the smaller subunit of the ribosome actually encounters the, yes, can you see this diagram, everyone? You all can see this. Let me just bring it here. Center will be good. Yes, this is visible to all of you. This is from your NCRT.
So,
Yeah. Now the actual mechanism starts when this smaller subunit, which you are seeing here, this is the smaller subunit of the ribosome, when this encounters the mRNA. Okay. So when it encounters the mRNA, it binds to the mRNA and starts reading the codes which are present on the mRNA. Now the first where it starts, every mRNA translation starts, is the start codon, right? And I told you that the first amino acid, first tRNA will carry AUG or will carry methionine and have complementary bases to the code AUG on it, right? Which will come and bind and start the translation. That is called initiator tRNA, correct? So write down the first point and we'll go point wise and I'll explain you the whole process. First point is when the small subunit encounters the mRNA, when the small subunit of ribosome encounters the mRNA, the process of translation of the mRNA to a polypeptide chain, an mRNA to a polypeptide chain begins. Okay. Now look here. There are these two regions. One is this, which I'm marking in blue. This is one site in the bigger subunit, and this is the another site in the bigger subunit. Can you see these two sites at which these two tRNAs are bound?
yes, sir.
Yes. So tRNA binds in the bigger subunit. mRNA binds in the smaller subunit. To bigger subunit. Now these two sites are known as A site and P site. Okay, A and P sites. This is not an NCRT and it's not very important to know the A and P, but just know that there are two sites. Now on the first site, the tRNA, the first tRNA comes and reads. So you can see here, look at this codon. in the ribosome, sorry, in the mRNA, it says GUU. Now, can you tell me what GUU codes for using the genetic code table? GUU. It codes for valine. Without going to the table, you can actually, yes, Mariam, it codes for valine. So see this tRNA, which is carrying valine here, has the complementary code CAA, which is complementary to GUU, right? So this comes, sits at this A site, okay, and binds on the complementary code and brings valine, because GUU, this code here, codes for valine, right? And when the two, in the last class, I told you about charging of tRNA, correct? What was charging of tRNA? Quickly. Charging of
Activation
tRNA.
of amino acid. Activation
Yes.
of amino acid in the presence of ATP
Yes,
during
Naveed.
linking to the specific RNA.
Yes, tRNA. So this valine here has some energy, okay, because it got activated in the presence of ATP. That energy, that charging of amino acid will allow that when these two amino acids, can you see alanine and valine are very close to each other here? Because the previous tRNA was carrying alanine. So this is very close to each other. When they come close to each other, there is a peptide bond formation between the two, because both of them have energy, right? So that energy gets stored in the form of a bond called peptide bond. So this bond here, you see, is a peptide bond, right? And this whole chain is known as the polypeptide chain. So there is a formation of peptide bond between the two amino acids when they are very close to each other. And the tRNA, which is sitting already on the site P, transfers the polypeptide chain to the tRNA, which is on site A. And then that tRNA goes away, flies off. Is it clear?
Yes,
So this goes away. And then when it goes away in the cell, it again, now, can you see that AGG? This is the anticodon loop. You can realize that this is the code of the anticodon loop, right? So AGG, what will be the codon it is reading? UCC, right? This one, UCC. You understand, everyone? It was reading UCC. Now, can you tell me what was UCC carrying? What does UCC codes for? UCC codes for serine. Yes? Yeah. And where is serine? See, this is serine. So this tRNA actually bought this molecule, right? Yes or no? So it bought this molecule, it transferred. So at one point of time, this tRNA was at position P, and this, which is now at position P, was at position A. So it transferred everything to the one at position A, then it left, and the one at position A shifted to position P. How it shifted? Because after you read the code, a tRNA comes and sits at the A. The tRNA at the P gives everything to the tRNA at the A and leaves. And then the mRNA shifts, like the ribosome shifts further. It goes here to read. When it shifts further, the next code will be AAU. So when it shifts further, the position A automatically becomes position P. You understand? This tRNA shifts to P. Is it clear? Then a new one comes, sits at the A, takes the polypeptide chain. The previous one leaves, and then it shifts at P. Then another new one comes, sits at A, takes the polypeptide chain. The previous one leaves, and this shifts to P. You understand? This keeps on happening until it reaches the stop codon, which is, what is a stop codon? Either UAA, UAG, or UGA. Yes.
Yes, sir.
When it reaches the stop codon, there is no tRNA that can now recognize. So there are some proteins that come and bind. Okay, these proteins are known as release factors or release proteins. They bind, stops the translation, and the polypeptide chain carrying tRNA, which is at the P site. So the A site is empty. Release factor comes and bind to the A site, and this whole polypeptide chain is released, and translation stops. So this is the process. Is it clear?
Yes, sir.
Yes. So keep writing now. Write down. There are, next point, there are two sites in the large subunit. There are two sites in the large subunit of the ribosome. In the ribosome. A and P. A and P, you can write it in bracket. Subsequent, next point, subsequent amino acids bind to these sites. Subsequent amino acids, or tRNAs, subsequent amino acids bind to these sites and come close to each other. And come close to each other. And come close to each other. So the close to each other, and the formation of a peptide bond, and the formation of a peptide bond takes place between them. Takes place between them. Okay. Is it clear, everyone?
Yes, sir.
Next point. The ribosome itself act as a catalyst, because ribosome has a RNA, right?
Yes, sir.
So that RNA, I told you in while teaching you RNA world, that RNA can also act as a catalyst, right? So how do ribosomes act as a catalyst? Because ribosomes have rRNA. So ribosome acts as a catalyst. You can write in the bracket that the rRNA acts as ribozyme, acts as an enzyme, ribozyme. So the ribosome acts as a catalyst for the formation of peptide bond. For the formation of peptide bond. Okay. Now, written, everyone?
Yes, sir.
Yes, sir.
Let's say this is the mRNA that was being translated. Okay. So it has a 5' end and it has a 3' end, right? Where is the start codon? Towards the 5' end. Because RNA is synthesized from 5' to 3' direction, RNA polymerase. And the stop codon is at the 3' end. But before and after this start and stop codon, there are some regions of the mRNA. Some additional regions. these regions are not translated and they are known as UTRs. So this will become 5' UTR and this will become 3' UTR. And UTR means UTR stands for untranslated regions. UTR, untranslated region. So which means that these two UTRs which are present before the start codon and after the stop codon are just for the kind of protect the mRNA and the information that it contains, right? But they do not get translated themselves. Is it clear, everyone, about the mRNA structure?
Yes, sir.
So write down. The mRNA has some additional sequences. The mRNAs has some additional sequences. Additional sequences present at the 5' end, present at the 5' end before the start codon and at the 3' end after the stop codon.
Sir, do the UTRs also have codons?
Yes, right. The UTRs, codons means they are one of the feature of genetic code is it is continuous, right? So if it is continuous, but you can only say, let's say the start codon, which is AUG, it starts here at this point, okay? And the stop codon, which is, let's say, UAA, is present here, okay? Understood?
Yes.
So before this, whatever is the code, it is the first. Where does translation starts from? AUG. The initiator tRNA can only read this one, right?
Yes.
So in the UTR, there will be no AUG. Okay. And in the 5' UTR. That's why the mRNA, when it comes and binds to the smaller subunit of the ribosome, no tRNA starts the translation because there is no AUG in this region. So it's not translated. So mRNA keeps shifting, and then when AUG comes, then the first tRNA comes and binds to the A region and starts the translation. Similarly, when it reaches the stop codon, UAA, no tRNA comes and bind, and instead a release factor comes and bind, and the polypeptide chain is already released. And now because release factor has bound already, whatever codes are after that, after the UAA, does not make sense. There will be, again, you need a AUG to again start translation, right? And there is no AUG in 3' UTR as well. Do you understand this?
Yes, sir.
Yes, Madhu? It's a very good question. So where was I in the dictation? What is the last thing you have written?
The mRNAs have some additional sequences present at the 5' end before the start codon and at the 3' end after the stop codon.
Yes, that are not translated. That are not translated, or which are not translated. Which are not translated and are known as untranslated regions, UTR. So they will have codons, of course, but it cannot be translated. Do you understood the reason why? Because start codon starts at a specific point after the UTR, and stop codon comes before the 3' UTR. Okay, everyone?
Yes.
Now, what is the importance of UTRs? Write down. UTRs are required. UTRs are required for efficient translation to happen, or efficient translation process. Why? Because you need some empty space. For example, there has to be, if this region contains some important information, this region is important, right? Because this region is the gene. Yes or no? We need some regions which are not important, which can actually be, while binding to the ribosome, these regions make the process more efficient. If you are making a painting, you always leave a border, right? There is a frame to any important photo or painting. So when you hold it from that frame or from the border, so that you do not touch the actual painting. You understand? Leaving a border makes it efficient to handle. Yes or no? Here also, leaving some untranslated regions before and after makes it efficient to handle. Cool. Now let's come to I think now write down, continue in the same point. Write down, for translation to begin. For translation to begin, the initiator tRNA recognizes the start codon. For translation to begin, the initiator tRNA recognizes the start codon, which is AUG. Followed by addition of amino acids. Followed by sequential addition. Write down. Followed by sequential addition of amino acids to make a long polypeptide chain. To make a long polypeptide chain and next point, last point, at the end, when the stop codon sequence is reached, at the end, when the stop codon sequence is reached, a release factor binds to the stop codon. First is initiation, where the
So can you
initiator
repeat after binds to the?
binds to the stop codon. So for initiation, initiator tRNA recognizes and binds to AUG. So the start codon. Then come elongation, where there is sequential binding of amino acids, one after the other. Okay. To form a polypeptide chain. The third is termination here also, where a release factor, basically it's a protein, binds to stop codon. And terminates the translation. And as a result, the complete polypeptide chain is released from the ribosome. Okay. Is the process clear to all of you?
Yes, sir.
Okay. Now we come to the regulation part of the gene expression, like regulation of the final protein formation. So this is translation. Write down. Regulation of gene expression. Now, if I ask you, at what levels do you think a gene expression can be regulated? So let's start with eukaryotes. So first thing, in eukaryotes, what do you think are different checkpoints where you can regulate the gene expression? You have studied the whole process now, from replication to translation. Finally, we made protein, and final protein formation is called the expression of the gene. Whatever was the gene, it is expressed in the form of proteins now in the cell. Simple. But at what levels do you think the regulation can be done in case of eukaryotes? What is the first initial level where you can do the regulation? In case of eukaryotes, you can start regulating the process or controlling the process at the transcriptional level itself. Right? So at the transcriptional level, where it is decided that how many mRNAs will be formed. You understand? So formation of primary mRNA. This is the first level. Suppose after you could not regulate it at transcription level. What you can do? mRNA has been formed. After that, can you regulate it at any other level? So at transcriptional level, we can decide whether we want the mRNA to be formed, how many mRNAs we want to be formed. We can control all of that, and hence we can control the gene expression. What if we did not control at this level? What is the other possibility? Let's say all the mRNAs that were supposed to be formed has formed now. Can we regulate it still? Madhya? Yes, no. Can we still regulate it, people? Yes, even if the mRNA is formed in eukaryotes, it has to undergo post-transcriptional modification, right? Processing. Yes or no?
Yes, sir.
So post-transcriptional processing, at this level, we can again regulate whether we want to process it or we do not want to process it, which means splicing regulation. If you do not do splicing, mRNA is of no use. Even if it is produced, it cannot make the gene expressed. So you can still regulate here. If you want to stop it, you can stop it by not splicing it. Suppose you have spliced also. Anything else you can do to regulate? Can you still stop the process or enhance the process? Splicing is done. mRNA is formed, splicing is done. Is there anything else you can do to regulate? What does eukaryotes have that prokaryotes do not have?
Membrane, nuclear membrane.
Exactly. So can we do something with that? Where is mRNA formed? Inside the nucleus or outside the nucleus?
Inside.
Inside.
So it has to go outside then, because ribosomes are present outside, right?
Yes, sir.
Which will translate it. So third regulation can be done at the level of transport of mRNA. Transport of mRNA from nucleus to cytoplasm. If you do not allow the mRNA to go to the cytoplasm, then does not matter if it is produced or it is spliced. If it does not go into the cytoplasm, it still cannot express, right?
Yes, sir.
And at the fourth level is the translational level itself. So somehow if you block the translation machinery, so at the translational level, you can again control it. So there are 4 levels at which you can control or regulate the gene expression in eukaryotes. Yes or no?
Yes, sir.
Yeah. Now talking about prokaryotes, tell me. I have told about eukaryotes, so you tell me about prokaryotes. At what levels do you think it can be regulated in prokaryotes? Do prokaryotes have post-transcriptional processing?
Yes.
No. Prokaryotes have a polycystronic gene. They do not have exons and introns. Remember? Forgot everything from the last class. I think that when you come to the class, you do not revise what was taught in the last class. And believe me, howsoever good a teacher teaches you, it's not enough for you to know things. Because if you just forget the best story that you heard, you just forget it. It's as good as not told to you. So if you want to take advantage from the way I teach, you have to revise on a daily basis. I have already told you that what is polycystronic and what is monosystronic, right? Only monosystronic gene arrangement undergoes splicing because there are exons and introns. In prokaryotes, there are no introns and exons. So why will they do post-transcriptional processing? Do they have a nuclear membrane?
No, sir.
So they cannot even regulate at the level of transport, right? They do not transport it basically. And I told you what is the characteristic feature of translation in prokaryotes?
A transcription translation both occur simultaneously.
Yes, they are coupled. Perfect. So there is a coupled transcription and translation, right? So the moment a mRNA is produced, it will start getting translated there only. So you cannot regulate it post the transcriptional through processing. You cannot regulate at the mRNA level. So translation and transcription both are coupled. So 1 and 4 are coupled in prokaryotes. So there is only one thing you can do to regulate. What can you do to regulate? You can only do one thing. By controlling the rate of transcriptional initiation. What was transcriptional initiation in prokaryotes? A sigma factor coming and binding to the RNA polymerase so that it can go and bind on the promoter region. Remember, people?
Yes, sir.
Yes, sir.
Yes. So that is the only place, only site, only level at which you can regulate. Once sigma factor has bound to RNA polymerase and it has bound to the DNA, mRNA will start producing. And once it goes into the elongation phase, you cannot do anything now, because as the mRNA starts coming out, it starts getting converted into a polypeptide chain. Is it clear?
Yes, sir.
So there is only one way it can be done. Now, apart from this, there is one. So in this transcriptional initiation site, there are some changes that prokaryotes do in order to regulate their gene expression. So write down this line. In the transcription unit of a prokaryote, in the transcription unit of a prokaryote, in the transcription unit prokaryote, the activity of RNA polymerase, the activity of RNA polymerase is regulated by the interaction with some accessory proteins. Some accessories proteins. Okay. These proteins affect the ability of RNA polymerase to recognize the promoter sites.
Proteins
Affect the ability of RNA polymerase to recognize the promoter region or the start sites. And third point, these proteins can act as, first they can act as activators. So some proteins are activators. Okay. So they have a positive regulation. Or they can be repressors, which have a negative regulation. Is it clear?
Yes, sir.
So what they have is, they have a different kind of a arrangement. They just add one more thing. What they do is, so I told you that there are other regions in DNA, remember, apart from promoter and terminator, which affect this whole process. I gave you one example as enhancers, right? Am I audible?
Yes, sir.
Am I audible, people?
Yes, sir.
Yes,
Yep.
sir.
Okay. My network was unstable. So instead of, apart from promoters and terminators, which are present on like beginning and end of the gene, and the structural gene is polycystronic, right? In prokaryote, remember that? There are some other regions known as the operator region. So let me pull out this image from your NCRT. Yes. Can you see this image, everyone? Let me bring it here. Can you see this? So this is how they regulate. So basically, this is called a Lac Operon. Lac word comes from lactose. Okay, this lac means here it is lactose dependent. What is lactose?
Sugar in milk.
Yes, it is a sugar which is present in milk. Now, this is a bacteria in E. coli. In bacterium E. coli, there is this arrangement. So can you see, this is, I think you can recognize this easily. See, this is a promoter region, right? This P is
Yes.
promoter. And then this is the structural gene. From here to here is the structural gene. And can you see one structural gene which is under one promoter actually is coding for 3 different polypeptide chain, 3 different genes, Z, Y, and A, right?
Yes, sir.
Apart from this promoter region, can you see there is this O thing here? This O, this additional O is called operator region. This is how they regulate their gene expression. Because once you start the transcription, there is no way you can do anything. In eukaryotes, you can do a lot of things. Even if transcription has happened, regulated post-transcriptional level, that has happened, regulated transport level, that has happened, regulated translational level. Here, the thing is coupled. So the significance of this operator region becomes very much. Now, how does it work? Just imagine as the operator region as You know, earlier, like when you used to go and stay in some hotel and someone wants to reach out to you when there were no mobile phones, and the person has to call the hotel first, right? And the call will go to a common operator, probably at the reception of the hotel. And the operator will ask, who do you want to talk to? And you have to tell that you want to talk to this person by this name who is living in your hotel. And then the operator will connect the line to your room's telephone line, right? Yes. So operator is a deciding factor. You understand? If operator does not allow or does not promotes the phone call to your room, you will not be able to continue with that call. You will not be able to talk to the person. Yes.
Yes, sir.
Similar thing operator does here. So there is two kind of regulations. This regulation is done through a repressor. So it is called a negative regulation. Lac Operon regulation. The Lac Operon is an example of a negative regulation. Here what happens, before this promoter, which will code for this sequence of the structural gene, there is another promoter. Can you see this is also a promoter, P region?
Yes, sir.
But what is this a promoter for? This is a promoter for this I gene. This gene. And this I gene codes for a repressor mRNA. And the moment this repressor mRNA is produced, it gets converted into a protein, which is called the repressor protein. And the moment this repressor protein is formed, it goes and binds to the operator region. It does not allow the operator region to be active. So it binds to the operator region, represses the operator region. Now, RNA polymerase, so let's say this is RNA pol, it cannot come and bind to the operator. You understand? Sorry, it cannot come and bind to the promoter. Because repressor is bound to the operator, it will not allow anything to bind to the promoter. In reality, these two regions, the promoter region and the operator region, are very close to each other. So when something comes and bind on the operator region, it shields the promoter region. This is the binding. Can you see this repressor molecule?
Yes, sir.
It is actually bound to the operator, but is also does not allow. Now this RNA polymerase, can it come and sit here? No. Right? It's not accessible. Is it clear, everyone? And this happens in the absence of inducer. Inducer is a molecule. In this case, lactose is the inducer. That's why it's called the Lac Operon. Now, what happens in lactose is that this lactose, if present, so this is case A. If there is no inducer, then this promoter will be active. It will synthesize the repressor mRNA. Repressor mRNA will produce repressor protein. Repressor protein will go and bind to the operator. And now mRNA polymerase cannot bind to the promoter. And this transcription will not happen. No transcription. So repressor binds to the operator region and prevents RNA polymerase from transcribing the operon. Is it clear?
Yes, sir.
Now coming to the scenario B, where inducer is present. Inducer is what induces the process. So this is the inducer, in this case, lactose. Okay. it comes, so again, RNA polymerase comes and bind to the promoter. So here also RNA poly will bind. It will synthesize the, transcribe the I gene, a repressor mRNA is formed, and a repressor protein will come out. But before this repressor protein goes and bind to the operator and make the promoter region inactive, this inducer, lactose, catches it midway. It binds to it. So inducer plus repressor makes the repressor inactive. You understand? Inducer induces the process but makes the repressor inactive. Now the repressor cannot go and bind. You understood? So now this binding is not happening. And if this does not happen, then RNA pol can sit here and can transcribe the gene, right? And when it will transcribe the gene, because it's polycystronic, there will be one single mRNA, which will be called Lac mRNA. But when it will be translated, there will be 3 different polypeptide chain. This is from the gene Z, this is from the Y cistron, and this is from A cistron. Remember, there are 3 different cistron. Cistrons are the regions that code for polypeptide. So 3 different polypeptide chains are formed. Is it clear?
Yes, sir.
Yes, sir.
Very good. Please make this, write all this information. This is a Lac Operon. You understood what is an operon? Yes or no? Sorry, one thing. Forgot it. Made a mistake. This inducer is not lactose. In the presence of lactose, this inducer is produced. So you can write instead lactose. So write down presence of lactose in the environment. So lactose is a sugar, okay, which bacteria can digest for nutrition. So presence of lactose induces the operon. Please note down whatever you need to note. If you can do it in your notebooks also, or you can just make it. It's very simple diagram.
Answer.
Done?
No, wait,
Everyone?
sir.
Yeah, take your time. Take 2-3 more minutes. I'm here on. Let me know when you're done. Done, everyone?
Yes, sir.
Others? Saifullah,
No, sir.
you're making it?
I'm in case B.
Okay. Let's finish it. Make it a little spacious. In your book, it's not spacious. So when you are making it with your own hands, in the process, make it spacious. Mark everything that I have marked here so that it is useful to you. Let me know when you're done, everyone.
Done, sir.
Done? Okay.
Yes, done.
Now, okay. Now write something about this operon. So what is an operon? If you have to define an operon, you can write down in the polycystronic structural gene in prokaryotes. Write down the polycystronic structural gene in prokaryotes
Polycystronic.
structural gene in prokaryotes. The polycystronic structural gene in prokaryotes is regulated by common promoter, and regulatory genes. A common promoter and regulatory genes. This type of arrangement is referred to as a operon. This type of arrangement is referred to as an operon. Is it clear? So basically, an operon is like a cassette. You have one starting point, but you can actually end up transcribing and translating multiple polypeptide chains. Now, why this happens? This happens because all these genes are not unrelated. They are related genes. Okay. So write down in a Lac Operon, you can see this minus I, this part here. Everyone, do you see this minus I?
Sir,
This is
what
known.
is the minus, sir?
So sorry, a small I. So
Yes,
small
sir.
I. Yeah, not minus I. It's a small I. So this small I is called a regulatory gene. Now, in this case, this regulatory gene is a repressive gene. It can also be a activatory mRNA. So in Lac Operon, or Lac Operon has one regulatory gene and multiple structural genes. In this case, there are three: Z, Y, and A. Multiple structural genes. All these genes present in one operon. Next point. All the genes present in one operon are needed together. To function in same or related metabolic pathway. Function together in same or related metabolic pathway. That's why they are coded together, because they are needed together. And it's a very brilliant, intelligent way to do this. Suppose in case of humans, we also have multiple genes which act together, but we have a monosystronic gene arrangement. If we need 4 genes to work together, we have to transcribe all those 4 genes separately. You understand, everyone? Because our structural gene is monosystronic. Do you understand this?
Yes, sir.
Because we have lots of space in the genome. Bacteria make it space efficient. So whatever genes are required, always required together, because they have less functions. They are a single celled organism. In our case, one gene can also affect multiple things, pleiotropic gene or polygenic inheritance. All these things also happen. So what they did is they put one promoter and multiple structural genes under it. So all these 3 are required together all the time. So they will synthesize it together all the time. Okay. Now just write down what is, sometimes they ask the functions of these genes. So write down Z, the gene Z, or the Z gene, codes for beta galactosidase. So overall, this Lac Operon works in doing what? Why it is called Lac Operon? Because this operon helps in the digestion of lactose, hydrolysis of lactose. So the Lac Operon, you can write down hydrolysis of lactose. This is what it does. See, so all the enzymes are related to that. So Z gene gives rise to beta galactosidase. And the function of beta galactosidase, write down, this is the primary enzyme. Primary enzyme responsible for hydrolysis of lactose. And second is Y gene. Y gene give rise to permease, another enzyme. This gene give rise to the enzyme permease. And the function of permease is, as the name suggests, permease. So it increases the permeability. What is permeability? Semi-permeable membrane permeability.
Ability to pass through.
Yes. So it increases the permeability of what?
Of lactose.
Of
Lactose.
yes, lactose. And or you can also write beta galactosides. Beta galactoside. It has lactose has this beta galactoside ring. Okay, so lactose also contains beta galactoside. So any molecule which contains beta galactoside, which can be broken by beta galactosidase. Remember, ase is an enzyme. Permease, beta galactosidase. So it increases the permeability to lactose and related molecules inside. And third is A gene. This codes for trans-acetylase, another enzyme. Now what does trans-acetylase do? What do you think trans-acetylase do? What does the name suggest?
Transformation of acetyl of
So what? Come again. Trans-acetylase, yes, trans-acetylase. What does it do?
Translates lactose into glucose.
It transfers an acetyl group. So it does not translates. It is not translational. It is trans-acetylase, acetyl group. So it transfers, it transfers an acetyl group. An acetyl group from acetyl coenzyme A. You have studied this in your 11th class. Remember acetyl CoA in respiration chapter, where you did glycolysis. If you have not studied this in detail, please go back to class 11. This is a very important metabolism part. From acetyl CoA, to lactose. So these are the three functions, the name of the enzymes and the gene. Gene, enzyme, and function. Please write it. So this is a lactose metabolic pathway in which all these three genes are required. Noted, everyone, written down?
1 minute.
Yeah, take it.
Answer.
Okay. Done, everyone? Shall I move forward?
Yes, done, sir.
Done? Okay. So we have studied today. We started translation. We studied the mechanism of translation, UTRs, gene regulation, eukaryotes, prokaryotes, and we have finished Lac Operon. This finishes the translation part as well. And now there are two topics left in this chapter. One is Human Genome Project, another is DNA fingerprinting, right? These are the two topics left. So we'll finish Human
Yes.
Genome Project today. So let's come to Human Genome Project. So what is Human Genome Project? Any idea about this? Human Genome Project. What does the term suggest you?
Finding the genes of a human.
How can we find the genes in a human? By finding out the total sequence, right, of the human genome. The full genetic material that we have, all the 23 chromosomes, 24 actually, because X and Y are different. So all the chromosomes and all the sequence of all the chromosomes. So Human Genome Project was a very big, it was a mega project. And you can imagine that how big was this project, because this whole project costed around $9 billion. More than that, but $9 billion was the funding for this project. Now, what was this project about? Write down. The Human Genome Project was a very ambitious project launched. It was a very ambitious project launched in 1990. That aimed at sequencing the complete human genome. Before this, some simpler genomes were already being sequenced, and we know we had the technology to do this. So we know what it takes to sequence a genome and to know what is the full genome sequence. So this project came out. Now, how big is a human genome? Human genome. Let's say if I talk about a haploid cell, any gamete, one copy, and how big is that? How many base pair are there? It has around 3.3 into 10 raised to power 9 base pairs in one haploid genome of human. Okay. You understood? Approximately. And the cost for sequencing one base pair was $3, around $3 for one base. If you have around, let's round it off to 3 for the sake of calculation. Even if you have 3 into 10 to the power 9 base pair, how many, what will be the total cost?
Nine into 10 power 9.
Yes, total cost to sequence complete haploid genome of human is equal to 9 into 10 raised to power $9, which is $9 billion. Understood?
Yes, sir.
And this is, if you do not understand how big is this, so just to give you the context, if you manage to sequence all the 3 billion base pairs, this is 3 billion base pairs, right?
Yes.
If you manage to sequence all the 3 billion base pairs, and then you decide to store the data in the form of books, if you store this whole data in form of books, and let's say each book has 1000 pages, and on each page you can write 1000 letters, A, T, G, C, like that. Then it will take 3300 books to store all the information from a single human cell, single haploid cell. You understand?
Yes, sir.
Yeah. So do you see how big is this data? And Human Genome Project was such a big. So can it be done? Can you do it manually? Is it easy to manage 3300 books and then analyze the human data to find genes and whatever you want to do with that? So can you do it manually?
No.
Yeah, because imagine a library containing 3300 books, and every book has just 1000 letters on each page and nothing else. No preface, nothing written, just it starts from a sequence, ends at a sequence. Suppose you want to find a gene, how will you find it? So what is the other way to handle such big data? Computers, correct? Yes or no?
Yes, sir.
So Human Genome Project not only made advancements in the biological studies, but it also developed the field of bioinformatics. Okay, so Human Genome Project led to rapid development of bioinformatics. You know what is bioinformatics? Computers in biology. When you handle data through computer software programs, or you make something with that field is known as bioinformatics. 3D modeling, annotating gene sequences, stuffs like that. Okay. Now about this Human Genome Project, you will be asked questions. So let's go to the questions that are asked in NCERT and let's go through questions. So the first question, write down. What were the goals of Human Genome Project? Such a big project cannot just start as it is, right? Someone has to propose the project. Yes or no?
Yes.
Anyone who will propose the project will be asked thousands of questions. Why do you want to do this? What is the benefit? How will you do that? What will be the funding? You know? So what are your goals? And the goals should be some real serious things, you know, otherwise no one will fund. And yes, before we go forward, you know who funded this? Who funded Human Genome Project? So just write down. It was funded by United States Department of Energy. And United States National Institute of Health. National Institute of Health. So these were the primary funders. They were the first two funders that funded it, funding agencies. After that, when they started the project, other agencies joined. For example, other countries also joined, like United Kingdom, Japan, France, Germany, and China. But these were the two important major funders. Is it clear? You can write down. After it started, countries like UK, Japan, France, Germany, and China joined in. Now coming to the question, what were the goals? So the first, the foremost goal was to identify all the genes present in human DNA. Which also means to find total number of genes. Okay. In your NCERT, I remember that if you go, do you have NCERT, everyone?
Yes, sir.
Just open this part where they talk about the goals of the Human Genome Project. There is one very, very silly mistake. Can you read the goals for me from the NCERT, Nabil?
Genome Project.
Chapter 6 towards the end. I don't know which
Yes,
page. But
identify
yeah.
all the approximate 20,000 to 25,000 genes in the human DNA.
Yes. Page 118, yes. Yes,
Determine-
yes, this, no, first, just read one. So they say that the goal was to identify all the approximately 20 to 25,000 genes. Now my question is, if human genome was being sequenced for the first time, how do you know that there were approximately 20 to 25,000 genes? No one has done it before, right? So you cannot know how many genes will be there. You can only make an estimate. And the estimate that scientists made and started with was much higher. They thought that human genome will have somewhere around 1 lakh genes. And even, it could be even more. This was what scientists thought. 1 lakh to 1 lakh 50,000 genes, approximately around 1 lakh to 1 lakh 50,000. Okay, so this first point is actually a silly mistake because they are telling you that they already knew there are 20 to 25,000 genes. No, it was not known at that time. And that's why these two big agencies funded it. They wanted to know how many genes and what is the total number of genes are there. And National Institute of Health is a health related institute which can then use this information for a lot of disease related conditions. So the actual goal was to identify all the genes present in human DNA, find total number of genes. Remember that. Second goal was what to determine the total sequence. To determine the complete sequence of human genome, which is around 3 billion base pairs. Clear? Then what if you find all that? What should be the next goal? Yes, Nabil? To store all the information in database. To make a database for human genome. Because you cannot store it manually. I told you what is the problem. It's very big, huge data. And once you store it, what now? Then you will develop tools for data analysis. So such a big data cannot be analyzed manually. Okay, so it can only be analyzed through computers. So you have to develop these tools. Then what after you develop the tools? Then you will share the information and the technology with the industries. Because everything at a broader scale has to be done in industries, right? You cannot do it in labs. You understand, everyone?
Yes, sir.
Any drug, a new drug is discovered in a lab, but is it synthesized and produced in a lab? It's produced in industries, right? Some company produces it on a large scale and then brings it out in the market. So transfer the related technologies to industry sector. And then someone also has to address the ethical, legal, and social issues. Because all the science is done by taxpayers' money, right? So you have to address the issues to the society that may arise from the project. Because people will ask questions, right? Someone will ask, what is it ethically correct? Is it legally correct to take someone's sample and sequence their genome? And what if you misuse the information? What if some scientists go and misuse the information of the genes? So all these issues have to be addressed. Is it clear?
Yes, sir.
So these were the goals. Now, how much of it was achieved? So as a project, some details about the project is Human Genome Project. It started in 1990, okay, and completed in 2003 on papers. So on papers, it was a 13-year-long project. But it actually got completed in 2006. Okay, so write down. The Human Genome Project got actually completed . 2006 in May. Okay. The last few sequences of chromosome number 1 was completed in May. Okay. Total, how many chromosomes total were sequenced? Tell me. How many chromosomes you need to sequence to get the total human genome? 24.
24.
22 autosomes because they are same, plus X and Y. They two are different, right? Yes or no?
Yes, sir.
Very well. Now, how was it done? So you don't have to, thankfully, you don't have to go into this detail of methodology because it's a whole chapter in itself. But I will tell you about the methods used in brief. I don't know if it's there in your NCERT or not, but you should know. So there were two major approaches. Two ways you can do it. Okay. First one is where you do expressed sequence tags. Is the process is known as expressed sequence tagging. In this process, what scientists do is that you focus on identifying all the genes that are expressed as RNA. So write down in this method, focus on finding all the genes, because if it is a gene, it will be expressed as a RNA because it will be transcribed, right? Yes or no?
Yes, sir.
It will tell us about the gene, only the gene sequence, but everything which is not coding for a gene cannot be known by this method. So this method identify all the genes that are expressed as RNA. Is it okay? So this way you can find the total number of genes, how many genes are there. But to find the complete sequence, including the non-coding sequence as well, because in our DNA there is a coding sequence and there is non-coding, like UTR, right? Untranslated region is not coding into protein, and there are many regions like enhancer regions or other junk DNA regions which we do not know what they do. They do not code for a mRNA or a protein. So for that, the other approach was sequence annotation approach. So basically what you do is, it is a blind approach. Write down, you simply sequence the whole set of genome. Okay. Second was sequence annotation approach. Remember these two words, expressed sequence tagging and sequence annotation. In this, what you do, sequence the whole set of genome. Sequence the whole set of genome in an unbiased manner, like blindly. Containing both coding and non-coding regions. Is it clear?
Yes, sir.
Yes, sir.
And after that, you are done this, then later assign functions to different regions, that which region of the DNA does what. So that is called sequence annotation. This is also known as blind approach. And this is express sequence tag. This is a specific approach, targeted approach. Okay. So how will you proceed? So these are the two major approaches, but how will you do it? Okay. So to do it, you will first take all the DNA total. So you will isolate total DNA from cell. Then you convert all the DNA into random fragments. Just break the DNA. And because you are randomly breaking, they will not have a specific place where they will break. They will break randomly in different sizes. Okay. Why is this breaking important? Because DNA is a very long polymer. You cannot sequence a very long polymer in one go. You need to break it down. Okay. Then you have to clone. Third is clone the fragments in suitable host. The host can be either bacteria or yeast. Yeast is a eukaryotic unicellular organism and bacteria is a prokaryotic unicellular organism. Both can be used as a host. And why do we clone? We clone to amplify each piece. Okay. When we clone, we amplify. So one piece becomes 10, 100, thousands in number. Then sequence the fragment. Sequence the fragments using automated computerized DNA sequencers. Okay. And these DNA sequencers work on the principle which was developed by Sanger, Frederick Sanger. This was the scientist who developed the principle for DNA sequencing, Frederick Sanger. And therefore, it is also known as Sanger sequencing method. Okay. So this is the overall, what can you say? Process. Now, talking about these suitable hosts, they are also known as vectors. So these suitable hosts can be of two types. Host vector for cloning. So these are the cells in which we clone the fragments of the human genome. It can either be done in bacteria. Okay. If it is done in bacteria, it is called BAC. Bacterial artificial chromosome. And if it is done in yeast, it is called YAC. Yeast artificial chromosome. Is it clear, everyone? Is this clear?
Yes, sir.
Yes, sir.
Yeah. Now, when it was done, everything which they wanted to find out was found. Okay. And what was found is asked as this one question. So there is another question about human genome that describe, write down this question, describe the salient features observed from human genome project, observed from human genome. Now the answer for this, you all have NCERT, everyone with you? Do you all have NCERT right now? People, yes, no? Come to page number 120. Your book. Quickly. Open. Do you see there are 9 features of the human genome written? Can everyone see it?
Yes, sir.
Yep. Let me just give me one moment. Let me try to pull it out for you. Okay. Yes. Yes. Can you all see this image that I have inserted?
Yes, sir.
Yes. Yes, someone please read the features and let me know if something is not understood. Who will read? Nabil? Nabil, are you there? Will you read these features?
Okay, sir. The human genome contains 164.7 million base
Yeah.
pairs.
Just simply you can write 3.1 billion base pairs. No need to remember this number. 3.1 billion base pairs is right. Okay. Yes. Next.
Average gene consists of 3,000 bases, but the sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases.
Yes. So normal average gene is 3,000 bases, but the biggest gene is dystrophin. Remember this name. It is important. Dystrophin has 2.4 million bases in just one gene. Okay.
Yes, sir.
Yes. Continue.
The total number of genes is estimated at 30,000, much lower than previous estimates of 80,000 to 1,40,000 genes. Almost all,
Yes.
which is 99.9% nucleotide bases, are exactly the same in all people.
So what was the estimate? 80,000 to 1,40,000, around 1 lakh to 1,50,000, which I said. And the total number of genes were found to be around 28,000. This is the approximate gene number in humans, which is less than 30,000. Okay.
Yes, sir.
Continue.
The functions are unknown for over 50% of the discovered genes.
Yes. So still we do not know the function for 50% of the genes. What is the function is not known. Okay.
Yes, sir. The functions are the less than 2% of the genome codes for proteins.
Yes, remember this. This is what I was talking about. Less than 2% of the genome code for proteins. Yep.
The repeated sequences make a very large portion of the human genome.
Yes. Sequence just repeat. Yes.
Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1,000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution.
So basically, this is just the same thing that above statement is saying, that there are many repeated sequences, and these sequences we thought that they do not have any direct functions. Okay, thought to have no direct functions, but in reality they shed light on chromosome structure, dynamics, and